Chapter 16. Lenski's Evolving Bacteria. Single-celled organisms, called Escherichia coli, exist in the billions in the guts of animals, including humans. They are usually helpful, sometimes neutral, and occasionally harmful. But in his lab at Michigan State, Richard Lenski has been observing E. coli without any medical goal in mind, and he has observed them continuously since February 1988. Bacteria divide, much as cells of the body divide. As they can replicate asexually every 40 minutes, Lenski and his lab assistants have seen a lot of bacterial divisions. The 60,000 generation mark was reached in April 2014. Today it is closer to 65,000. The experiment is supported by a grant from the National Science Foundation. Lenski calls it his long-term evolution experiment. His underlying argument is that even in the absence of selection pressure, E. coli will evolve on their own, given enough time or bacterial divisions. A member of the National Academy of Sciences and the Hanna Distinguished Professor of Microbial Ecology at Michigan State University, Lenski has focused on genetic mechanisms that drive evolutionary change, his lab reported. 25 years of E. coli divisions are said to be the equivalent of 1 million years for humans. Others say it is closer to 1.5 million years. Well-known science journals soon became Lenski's greatest admirers. Elizabeth Pennisi's article about him in Science, published in 2013, covered 4 full pages of the magazine. It was headlined, "The Man Who Bottled Evolution," with this subhead: Richard Lenski's 25-year experiment in bacterial evolution shows no signs of running out of surprises about how mutation and selection shape living things. Lenski and assistants grow bacteria in a dozen nutrient-filled flasks. Then, every day, 1% of the population of each flask is transferred to a new flask of fresh growth medium. Under these conditions, each population experiences 6.6 generations or doublings per day. The flasks are kept separate from each other so that the 12 different lines of descent don't get intermingled. Richard Dawkins has called the separate flasks tribes. About every 500 generations, taking about 8 weeks, Lenski and his helpers deposit bacteria from each flask into a freezer. These become the ancestors. They can be thawed whenever needed and then compared to their most recent descendants. In this way, Lenski has created a fossil record of his bacteria. The experiment continued along these lines for over 25 years. Carl Zimmer, a journalist and the author of Evolution, The Triumph of an Idea, 2001, reported in Discover magazine that the bacteria have been evolving in all sorts of interesting ways, and the freezing and thawing has allowed Lenski to reconstruct the history of that evolution in great detail. The experiment is an absolutely magnificent achievement, said Douglas Fatuyma, an evolutionary biologist and textbook writer at Stony Brook University. Richard Dawkins said that it is a beautiful demonstration of evolution in action, something it is hard to laugh off even when your motivation to do so is very strong. And for creationists, Dawkins added, that motivation is very strong indeed. But before we get carried away, now may be the time to mention what is easy to lose sight of, that after so many generations and so much evolving, the bacteria remain bacteria to this day. It seems likely that they will stay that way for as long as the experiment lasts. It has no defined endpoint, says Lenski, but he wants others to keep it going after he retires. He would like to see his experiment continue for another 25 years. The biochemist Michael Behe told me that the experiment can show you what can happen in long-term evolution. In his 2010 article on Lenski's experiment in the Quarterly Review of Biology, Behe pointed out that when trillions of bacteria replicate over 60,000 generations, numerous mutations are to be expected. Yet the most important evolutionary changes observed so far have involved the loss of old genetic functions rather than the acquisition of new ones. Behe said that he disagreed with Lenski's interpretations, but nonetheless he is a big fan of his work. He explained why. Although Lenski is decidedly not an intelligent design proponent, His work enables us to see what evolution actually does when it has the resources of a large number of organisms over a substantial number of generations. Rather than speculate, Lenski and his co-workers have observed the workings of mutation and selection. For this, we intelligent design proponents should be very grateful. In his 2007 book, The Edge of Evolution, Behe said that unlike other microorganisms, which have to fend for themselves and contend with the human immune system, Lenski's bacteria have been coddled. One might say that they are on welfare. They have food, a stable environment, and no predators. But doesn't evolution need a change in the environment to spur it on, Behe asked? And he further asked, shouldn't we expect little evolution of E. coli in the lab where its environment is totally controlled? Behe answered both questions, no and no. One of the most important factors in an organism's environment is the presence of other organisms. Even in a controlled lab culture where bacteria are warm and well-fed, the bug that reproduces fastest or outcompetes others will dominate the population. Like gravity, Darwinian evolution never stops. So Behe would concur, at least in part, when Lenski said in a National Science Foundation press release, "Most of us think about evolution in the past tense. After all, we were first exposed to the concept of evolution when we saw dinosaurs and other fossils at museums. But evolution has not stopped. It is an ongoing process. Charles Darwin emphasized this point when he closed on The Origin of Species by saying, 'From so simple a beginning, endless forms most beautiful and most wonderful have been and are being evolved.'" Behe agrees that evolution can be thought of in the present tense. But of course, the big difference between Darwin's theory and Lenski's experiment is that Lenski began with E. coli in his flasks, and that is what he still has over a quarter of a century later. Yes, there have been some changes. A year or two after the experiment began, something happened within the flasks, making the bacteria reproduce faster and take over the flask they were growing in. So there was a mutation, and it conferred a survival advantage on the bacteria. Lenski would say that was just what Darwin would predict. Then later on, here comes another mutation, and that helped even more. That was true enough, and it attracted headlines in the newspapers. But what changed exactly? The problem initially was that Lenski had no way of knowing the molecular details of those changes. All he knew was that the bacteria would grow faster. Also, the average size of the E. coli increased. Then in about 2005, new genome sequencing technology made it possible to read the DNA letter by letter. Today, the entire genome of E. coli has been sequenced, and the frozen record of Lenski's ancestral bacteria has allowed comparisons to be made between the bacterial old-timers and their most recent descendants. Unlike humans, bacteria don't mind being frozen, then thawed, and studied all over again. But once Lenski's lab did identify the mutations at the DNA level, many of the beneficial mutations turned out to be degradative ones. They were breaking or deleting pre-existing genes or regulatory elements, meaning that those genes or elements no longer worked. This is something that Lenski's enthusiasts, including Richard Dawkins and Jerry Coyne, the latter in Why Evolution is True, have failed to point out. Lenski himself is inclined to emphasize the positive in his experiment and to downplay the degrading of genes. Sometimes a broken gene can actually help an organism, under the conditions in which it was grown, Behe pointed out. He gave this example. One change that helped the bacteria grow faster was that they lost their ability to make flagella, so the bacterial flagellum was essentially thrown out. The reason was that in his flasks, the bacteria were shaken while they were growing, so no bacterium had to swim. Why spend energy on something when you can get a free ride? But that does not explain where the flagella came from in the first place. From this, Behe formulated his first rule of adaptive evolution. Break or blunt any functional coded element whose loss would yield a net fitness gain. He also emphasized the huge difference between constructing something and demolishing it. Blowing up a bridge is not the opposite of making one, he said. In fact, knowing how to demolish something tells you little or nothing about what was needed to build it in the first place. That was one of the very interesting things, and it wasn't just the flagella. A number of other genes were not needed anymore because all the nutrients that the bacteria needed were supplied in the flask. So the ones that were needed in a normal environment were broken and thrown away. In a paper in the Proceedings of the National Academy of Sciences, Lenski gave another example. One of the mutations removed the ability of bacteria to proofread their own DNA. Normally, mistakes get incorporated into DNA, but as we saw in the previous chapter, cells have mechanisms to spell check a text and fix them where the mistakes are made, as a typist corrects a typo. But the bacteria in Lenski's flasks lost the ability to do this. However, the cells that had the mutation causing the loss of proofreading ability were growing just as fast as the previous cells that had the original proofreading ability. The question then was why the loss of proofreading ability didn't hamper the growth of the cells. When Lenski looked more closely to see what had caused this, it turned out that yet another protein had been lost by another mutation, and this loss had a compensating effect. Behe summarized the outcome this way. The initial mutation was the loss of one kind of protein, the kind that helped proofread it, and the second one, compensating for the loss of the first one, was the loss of a second protein. And that serendipitously helped. But you're going to run out of proteins to lose in a fairly short order if you keep on going like that. The most important claim, reported by Elizabeth Panisi and others, was that the bacteria had evolved a new way to feed themselves. Instead of relying on glucose, they drew on a different energy source in their medium, citrate, which enabled them to reach much higher densities than in other flasks. This was the biggest event in the entire E. coli experiment, said Lenski's assistant Christoph Adami. To have a complex new function develop seemingly from scratch is a big deal and quite remarkable. There was a lot of citrate in the medium, and in the absence of oxygen, the E. coli have no problem consuming citrate. But when oxygen is present, as it is in Lenski's experiment, the bacteria can't digest citrate. Later, however, they found that a fairly simple change made its digestion possible. Citrate then became the new food supply, and that allowed the mutated citrate-consuming bacterium to outcompete its relatives, Behe wrote. The ability to consume citrate became visually conspicuous in one flask at about the midpoint of the experiment, after about 33,000 generations. The normally clear flask became cloudy with the profusion of bacteria on citrate. But at the molecular level, nothing novel occurred. For as Behe explained, the change was relatively simple. A piece of DNA close to the citrate gene got duplicated by accident. That put a control element next to the citrate gene, which allowed it to be active in the presence of oxygen. Nothing novel occurred because the control element already existed beforehand. All that happened was a duplication of what was already there and a rearrangement of the positions of the gene and control element. The analogy of a single-lane highway becoming a two-lane highway has been used to account for the ability of the bacteria to consume citrate. And yet, I asked Behe, doesn't the building of a whole new highway lane seem like a fairly complex change? In response, he developed the analogy further. It is as though the highway lane were already there, he said, but blocked by a sawhorse barrier. The mutation essentially knocked the barrier away, and traffic could then use the pre-existing lane. Behe said that the Lenski experiment had failed to show beneficial mutations that resulted from building what he called new functional coded elements, FCTs. Roughly, that is a sequence of DNA that helps in the production or processing of a gene or a gene product. Improvements had been made by breaking existing genes or fiddling with them in minor ways. But new genes or regulatory elements still had not shown up. Richard Lenski was interviewed by National Public Radio for its morning edition in 2013, celebrating the 60,000th birthday of the bacteria. Random mutations had allowed the bacteria to improve their fitness, meaning to reproduce faster. In evolutionary biology, Lenski told NPR, fitness is this representation of the ability of an organism to survive and reproduce. All else being equal, Organisms that reproduce more quickly will have an advantage when competing with those that reproduce more slowly. Readers may recall that Lenski brings us back to the same problem I began with at the beginning of this book. What does fitness mean? Lenski defines it in terms of faster reproduction. But the problem is that surviving even as you reproduce faster is not quite what Darwin had in mind when he talked about the mechanism, natural selection, that is said to explain the origin of species. In 1966, the geneticist C.H. Waddington said that some of the statements of neo-Darwinism are vacuous. Darwin's understanding of fitness was later turned into a lot of mathematics, Waddington added, and then it was redefined, by Ernst Mayr among others, simply in terms of leaving more offspring. Therefore, Lenski leaves us in the same place that Waddington criticized. Some E. coli, if left to their own devices, leave more offspring than others in a given time period, and that is all there is to it. The idea is smuggled in, to echo Waddington, that the bacteria leaving the most offspring are going to be the best. But nothing like this is explicit in the theory. Similarly, all Lenski has shown is that some organisms leave more offspring than others. Michael Behe summarized the experiment this way. Lenski's lab did an immense amount of careful work and deserves much praise. Yet the entirely separate $64,000 question is, what do the results show about the power of the Darwinian mechanism? The answer is they do not show it to be capable of anything more than what was already known.